We're here to talk about a project that we've been undertaking over the last couple of years now, isn't it? Looking at the dietary impact of the Norman Conquest, building very much on the work that Naomi did back about 10 years ago. It was published, wasn't it, 2007. And what's distinctive about our project is that it's a multi-proxy and multi-scale study of diet, which means that we're combining stable isotope analysis at bulk level, but also incremental stable isotope analysis, which Lizzy will go on to talk about in a moment, which is being undertaken by my colleague Richard Madrick at Cardiff. Organic residue analysis of ceramics, which has been undertaken by Lucy Cramp and Helen Welton from the University of Bristol, and osteology, which has been led by Lizzy. So what we want to be doing is looking at to see if we can see changes in diet at different scales from the individual through a single life course through the osteology and the incremental isotope analysis up to a sort of fuzzier picture which the organic residue analysis and the bulk isotope data provides. For those of you who maybe why study diet type question, it's really food is at the intersection of society. It brings together environment, changes environment impact the foodstuffs which are available to eat, economy, how we get hold of those foodstuffs, how they're marketed, social identity and culture. So those obviously input a great deal onto the decisions that we make about what to eat and also how to eat it. So food really is the nexus of all these different things, and I guess much like the parchments that we've been hearing about already, they provide a focus through which we can bring together all these different elements of social, cultural, economic change which we can see going on in this period. So I mentioned already that Naomi has previously studied the zoo archaeology of the Norman Conquest. I don't know why our bullet points are appearing as question marks. The headline finding, I think, which comes out of Naomi's work is that after the Norman Conquest we're seeing increasing pig consumption in England, possibly mirroring a pattern which is already established in France, the introduction of new breeds and also a focus in particular on eating younger pigs. But this is something which is very much emphasised at elite sites. We're also seeing agricultural intensification and an increase in wild animals and exotica, again mirroring principally the elite taste for hunting. Work by Alex Levada looking at archaeobotanical evidence has demonstrated an increase in the availability of certain condiments. It's worth saying that this probably isn't directly related to the Norman Conquest. Much like the fish event horizon of 10,000, it's probably linked to, not 10,000, 1,000, 1,000, 1,000, linked to increasing urbanisation, increasing international trade links, which could have been catalysed in an English context by the Norman Conquest. My own analysis of ceramic use in Southampton, showing the development of new cooking styles, principally, seemingly we're seeing slower cooking, not using such heat, which is something which we'll come back to, I think, in a moment. Possibly in a port, seeing mixture of English and French styles. I mentioned the fish event horizon already. Around 1,000 we see a massive spike in the consumption of fish. And all of this has been brought together in a paper in the Archaeology of the Norman 11th Century volume, edited by Chris Dyer and Dawn Hadley, which came out at the SMA conference a few years ago, in a paper by myself, Alex Levada, and Fiona Whelan, who's done all sorts of interesting stuff looking at written sources about food culture, which unfortunately we haven't really got time to go into. But the headlines really of what we know so far are that we're seeing a marked change in elite food culture, but we don't really know what the impact on ordinary households was. And that's really what we set out for this project to find out. So we were successful in getting some small grants from the Society of Medieval Archaeology, the Royal Archaeological Institute and the Society of Antiquaries, all three January deadline funding applications. We decided to focus on the city of Oxford for a pilot study to see if we could look at an urban population and combine those different methods which I mentioned already. And the principal reason that we chose Oxford was that it has some nicely well-dated human remains that we could use. So Oxford is a crossing point on the River Thames. It's a really interesting town from a political perspective because, of course, the River Thames was that important boundary between the Viking Danelaw and Wessex. In the 8th and 9th century, there seems to be a minster in Oxford, dated in part by the presence of Ipswich ware pottery and also some radiocarbon dates, founded as a burh by Alfred in the 10th century. We've got historical documentation and lots of archaeological evidence for the building up of the street frontage. And then after the Norman Conquest, the castle, which is the blue blob, sort of turquoisey blob on my side of the screen here, displaced some Anglo-Saxon buildings and also seemingly displaced a Anglo-Saxon or pre-conquest burial ground as well. So now Lizzy's going to take you through some of the techniques and findings.
Okay, so moving on. So Ben's already mentioned the fact that Oxford provided an abundance of well-dated material, and this allowed us to break down the chronologies, in some cases into pre-transitional and post-conquest. In other cases at the moment we're just working with a pre- and post-conquest division, but really that's a key aim of trying to unpick the chronology of what's going on in Oxford. The aim here was to try and have a more nuanced understanding of dietary change. We wanted to focus on data that didn't derive primarily from the elite classes, and we also wanted this opportunity to bring in this MULTI-PROXY study by way of trying to shine a more complex light on what's going on, but also by way of trying to see whether a MULTI-PROXY study would actually answer the questions that we wanted it to answer in the first place. So as Ben mentioned, we've got organic residue analysis of ceramics, 2 different kinds of isotope analysis, which I'll explain as we go along, and a focus on human osteological data, primarily dental and skeletal pathology. So to begin with the organic residue analysis, we analysed 41 sherds, of which 68% hit the threshold of 5 micrograms to the gram, which is the cutoff for preserved lipids. So we ended up with 28 data points in this data set. It comprised a mixture of dated local wares. They were from a range of different sites, and it wasn't simply the case that the same pottery came from the same sites. It really was a bit of a mixture. And that broke down into late Saxon Shelley ware and Cotswold ware, both of which are pre-conquest in date. The St Neots ware, which spans the conquest, is 11th century, and Oxford ware, post-conquest pottery. So if you have a look at the plot, essentially what happened here is that the data was presented by pottery type. So the 4 pottery types are separate charts, but I'm going to think of them all kind of combined together. The first pattern of note is the use of these pots for various kinds of fats. Ruminant dairy fat use seems to be limited to the pre-conquest sample. We've got 1 sherd indicating a very high level of use of this, no use for any other fats. In contrast, we have predominant use for non-ruminant adipose in the post-conquest pottery. I'm not sure I'm going to be able to make this work. I won't bother. The blue circles show you 2 examples of Oxford ware, which were, in terms of fat use, predominantly used for non-ruminant adipose. Moved on. Okay. So ketones, indicating prolonged heating, were detected in 5 sherds. These are all pre-conquest Shelley ware. And it's notable there's no similar evidence for high temperature heating in our post-conquest wares, or indeed in the pre-conquest examples of other ware. We also found evidence of compounds typical of brassica, in particular leaf wax, so essentially cabbage, in a variety of pot sherds of various types and various dates. Although brassica might seem a little bit more common in the post-conquest sherds, it's notable that it's found in combination with the same sorts of animal fats across different pottery and is not associated with any particular site type. Finally, 1 example of beeswax in just 1 pre-conquest sherd, and no evidence of tars or resins in any of these particular samples. So several potential interpretations could be interwoven with some of the stuff we know about what might be going on during this period. We've got ruminant dairy fats appearing more common in the pre-conquest sherds, and non-ruminant fats more common in the post-conquest sherds. This can understandably be tied into what we might be thinking about what's going on with the exploitation of pigs. There's no evidence for intense heating of animal fats in our later sherds, and there is this association as well with the Shelley ware. Both the pottery type and the practice of high-level heating seems to disappear at the same time. And we've wondered about whether that kind of ties into a shift in not only what's being cooked, but the way in which things are being cooked. So perhaps in association with the introduction of practices such as roasting. We do have to think more broadly about how some of these practices might tie into what's going on in Oxford, and certainly ethnic differences in cooking practices have been raised before. The idea, for example, that Shelley wares were in core use in the Saxon heart of the burh, whereas perhaps the St Neots ware was peripheral and used by Danish populations, Oxford being perhaps quite a complex place at the time. So we'll have to tie into some perhaps more nuanced thinking as we develop this project in the ways in which and different people who were using these different wares. And cabbage use is consistent across wares and with fats, so whatever's happening, people still like cabbage. Moving on to look at some of the isotope analysis. We're going to begin with the analysis that was undertaken on bulk bone collagen, which includes 36 humans and 60 animals of pre- and post-conquest date. One of the most noteworthy patterns to pick out here when we compare the phased human samples, and the humans are on the far side, circled by the large coloured rings, is that we seem to have a lot more diversity in pre-conquest diets than we do in post-conquest individuals. The standard deviations are far higher in the pre-conquest sample in both carbon and nitrogen. Now, thinking about this reduction in variability in diets would fit quite nicely with a pattern where we see increased control of market supply in Oxford under the Norman administration, increased urbanism, perhaps tighter controls on agricultural production and supply. It's plausible here that the post-conquest communities had a more homogeneous diet and that the supply was more stable in order to facilitate that. In addition, it's worth pointing out what's going on down here with this black dot, which is our post-conquest pigs. They are notable compared with the other animal samples for having, again, a narrower standard deviation range. This is particularly noticeable in the nitrogen. So if we combine this with evidence of increased urbanism, it's tempting to start thinking about what the way these pigs were being managed. So if they were being kept in the confines of the city as sty pigs, for example, being fed on waste from the increasingly growing urban population, then we might see these sorts of patterns occurring in the data. And indeed, if the zooarchaeological evidence suggests pig consumption increased markedly after the conquest, then this reduced variability in human values might also relate to the consumption of this more homogeneous pig protein. Moving on to look at the incremental data, just by way of a brief introduction for those of you who might not be familiar with this sort of analysis, instead of producing a single value per individual, our incremental isotope data tracks dietary change across childhood. It does this because we take sequential samples through a tooth, thereby tracing dietary change and indeed aspects of health across the period when that tooth was developing. So we looked at 9 individuals. They were all of the sort of transition post-conquest period by nature of the samples. We couldn't look at any more, unfortunately, because the amount of analysis involved in this technique requires quite a lot of money. So we ended up looking at second molars of 9 human individuals. This tracks the dietary and health change between 2.5 and 14 years-ish of life. If we have a look at some of the variation here, I think what we're presenting at the moment is a bit of an insight into how this technique might provide a kind of higher resolution insight into some variation of diet across the life course. With this amount of data, we really can't say too much about bigger changes within Oxford. So the first individual shows really quite stable diet. We have carbon values and nitrogen values, both of which seem relatively stable. And this is notable because nitrogen values, they do vary a little bit, but they're sensitive to diet and health. And so we might be able to draw some conclusions here about the stability of this person's lifestyle and experiences. Indeed, there's less than 1 part per million difference in nitrogen across the length of the tooth. So we're suggesting very little change in diet, but perhaps very little change in overall health variation as well. And it's notable that this individual has quite high nitrogen values across the board, which would suggest a reasonable consumption of animal protein consistently through their developmental years. If we contrast that with someone who's had a very different dietary experience, this individual's from Oxford Castle, they show far more marked covariation of nitrogen and carbon, and the nitrogen having a range of up to 5.5 parts per million across the tooth axis. And this is a much more unstable diet here. Values of less than 7 parts per million are beginning to suggest this individual has limited intake of dietary protein at certain points during their life. So we're seeing variation here, we're seeing individuals that are experiencing stable and unstable diets. With this small amount of data, there's not too much we can say more generally, but we do feel this approach has quite a lot of potential to drill down into some of the personal experiences. And given the fact that political and social instability across the conquest could very easily have been very short term in its effects, this might provide a way of investigating this a little bit more. Moving on to the human remains, finally, we wanted to explore diet and health, perhaps using slightly more traditional techniques and a very well available data set for Oxford. We looked at all of those individuals sampled from isotopes and many more, and built up a data set of 254 individuals for this stage of the analysis. Now, it's worth pointing out here we're looking at some new analysis that we undertook ourselves, plus an awful lot of data that's been obtained from grey literature and site reports, and that did, to a certain extent, limit what we could and couldn't do in terms of comparative analysis. There is a sex imbalance in this data set. Unfortunately, there are much fewer women among the pre-conquest sample. This is something we're going to have to consider quite carefully through the more nuanced stages of interpretation. And it's worth pointing out that this is quite an exploratory way to think about diet, because the etiologies, the causes of the various pathologies that we can look at in human skeletal remains, are not fundamentally diet. They reflect diet, but they do so through a series of proxies. Having said that, there were some quite consistent patterns we found in the data. We find higher levels of dental pathology, particularly calculus and periodontal disease, in our post-conquest sample. Calculus is a bit problematic. Bioarchaeologists tend to associate it with high protein diets, which would be wonderful, but it is also associated with high carbohydrate diets, with the coarseness and consistency of your diet. The coarser the diet, the faster plaque wears away, so calculus doesn't build up so quickly, and a range of complex genetic factors as well. Nonetheless, we do see an increase in our post-conquest individuals. And we also find a higher rate of a suite of pathological conditions that are associated with, for want of a better term, biological stress. And these are the pathologies that are often associated with periods of dietary and social change. However, things become a little paradoxical. First of all, we have to point out that we don't expect the Norman conquest in Oxford to have acted directly to reduce overall population health. Documents suggest that land was laid to waste in Oxfordshire to thwart any Midland resistance there might have been, but Doomsday suggests the impact was probably fairly short-lived. So we're not looking at a town where the kind of basis of the subsistence economy was destroyed. There is also increasing evidence from those working on these kind of pathological lesions to suggest we have to bear in mind that a high prevalence of stress markers might actually, paradoxically, suggest a population that's doing reasonably well, particularly with linear enamel hyperplasia, which you see on the far side, which is statistically more common in post-conquest individuals. It probably indicates people that are actually surviving through childhood stress insults. And this is the sort of thing that would be consistent with a more developed urban economy, a more stable situation in terms of access to food resources. This is what might well manifest in what we're seeing here in the population in Oxford. Can we pass over?
Okay, so to wrap up, we have been using these different techniques then to try to come up with a sort of multi-scalar approach to thinking about diet and health. And actually it's the health side of things I think I've got a bit more interested in and excited by than actually the what were people eating side of things. So we do seem to be seeing across all of the evidence this evidence of a more stable diet, possibly to do with the maturity of the market system, contrasting to the diversity of the pre-conquest period, and whether or not the extent to which the conquest is to blame for that, as it were, is up for debate, because of course urbanisation and commercialisation was a longer term process than that. We are seeing the pigs, and we're also seeing potentially change in husbandry practices of the pigs, as Lizzy mentioned, and potentially we're seeing this change in cooking practices as well, although we do need to be quite cautious because the data set's so small and also quite nuanced given the social complexity of Oxford, particularly in the 11th century. We are again seeing that little bit of variability between the individuals, and actually when talking about calculus, something twigged in my mind about Matthew mentioning Anita Rodini's work on calculus, and it'd be really cool if you could throw that into the mix as well when you're thinking about the biographies of these people. So quite what the increased stress markers are showing is again a bit blurry at the moment, as Lizzy said about these paradoxes. But if we are seeing short-lived dietary stress around the mid-11th century, then that's perhaps what I would actually expect to see rather than a prolonged decline. Certainly in the bulk of evidence for Oxford, you don't really see a sort of material trace of the conquest as such. So to sum up, really, in developing this approach, I think we've highlighted quite a lot of potential, and there may be areas where this potential could be better exploited than others, but also, of course, as ever, a great deal of interpretive problems to overcome as well. So there we go. Oh, and finally, we have to thank all the money people.